neuropathologist. Zane is an academic neuropathologist based at the Division of Neuropathology and the Queen Square Brain Bank. She is actively involved in both clinical practice and research in neuropathology, with a special interest in neurodegenerative diseases associated with misfolded protein aggregation. So thank you for joining us today, and I will hand over now to you, Zane.
Thank you very much, and good morning, everyone. I'll share my screen. I think a request has been requested for sharing the screen.
And please do say your name for me, because I'm sure I butchered it.
Yeah, good morning, everyone. Would you just mind confirming that you can see my screen?
Yes, we can. Thank you.
Yeah, thank you. Yeah, good morning again, and thank you for the very kind invite. So my name is Zane Jaunmuktane. As already kindly introduced, I'm a Associate Professor and Consultant Neuropathologist at Queen Square Brain Bank in London, and I am presenting our work on behalf of my colleague, Patrick Cullinane, who is a clinical neurology research fellow. So I'm a neuropathologist and he's a neurologist. And he really wanted to be here today, but he had to be in the clinics with the patients this morning. So that's why I'm presenting our work on his behalf, but we are working very closely together. And what I will talk you through today is our research in primary tauopathies, specifically focusing on progressive supranuclear palsy, and what we really are interested in. And very just to set the scene and very quickly explain the background. Obviously, primary tauopathies, they consist of quite a number of diseases, from which the commonest ones are called corticobasal degeneration and progressive supranuclear palsy. And the reasons why these diseases develop, we obviously do not know, unfortunately, as of today, but there are a few things we do know about these diseases. One thing we know that these diseases develop if the normal functional tau protein, shown here in a neuron in orange color, if it changes for some reason its shape to a pathological form and starts to clump together with other pathological forms of this abnormal dysfunctional tau protein. And then if in a given cell, in an astrocyte, which is a supporting very important cell in our brains, if it deposits, it clumps in these big clusters at the distal ends of the processes, then a disease, what develops, is called corticobasal degeneration. But if for some reason it clumps in these proximal processes close to the cell body, as shown here, then the disease which develops is called progressive supranuclear palsy. We increasingly are also recognizing that in all primary tauopathies, either progressive supranuclear palsy or corticobasal degeneration, actually there are additional co-pathologies present in patients' brains. And these pathologies, these additional pathologies, they can either be of Alzheimer's disease type, where there's a combination of amyloid beta dysfunctional protein together with a different kind of tau protein, or it can be of Parkinson's type, where there is deposition in addition to this abnormal tau protein, also Parkinson's type protein called alpha-synuclein. And also there can be another protein called TDP43. And patients also frequently have concomitant systemic pathologies such as heart disease or diabetes. And this combination of these other pathologies, we are increasingly recognizing they might contribute to the progression of the disease, but to what extent, we do not know at this stage. But at Queen Square Brain Bank, together with Patrick Cullinane, we're very much interested in understanding the basis and how these co-pathologies influence the primary disease progression. We think that disease progression, because of these co-pathologies, could be, the symptoms could be worsened, and the spread and the pathology deposition could happen at different speeds. There is also a possibility that what we know that patients can have different unusual symptoms, that these co-pathologies could influence these unusual atypical symptoms, and that all will have a significant relevance for identifying a biomarker, a diagnostic tool, how we can diagnose these diseases early in disease process accurately, and will be important for correct interpretation of these biomarkers of these test results. And then inevitably, once eventually there is a disease-modifying treatment, these co-pathologies might show different response to the treatment. And together with Patrick at Queen Square Brain Bank, we really much are interested in understanding the role of co-pathologies in primary tauopathies. We are very much interested in understanding whether focal specific molecular cell specific changes in a very specific anatomical brain region might also influence the development of unusual symptoms in patients with primary tauopathies. And by far most importantly, we are very much interested in using human tissues, helping identifying early diagnostic tools, how disease could be diagnosed early and accurately. And we particularly are interested, and that's what work we are doing with the support from PSPA Association, is looking at protein modifications as a potential tool of diagnosing these diseases early. And what I'm going to talk you through today is about all our ongoing work, some of which is supported by PSPA Association, where we are using computational assistance, artificial intelligence, to look at the pathology, to look at the clinical notes, to look at gene activity in very specific cell types, and also to look at these protein modifications as a potential tool of early diagnosis of primary tauopathies. So what we have curated from Queen Square Brain Bank collection is over 600 brain donations, of which majority are from patients who died of PSP, progressive supranuclear palsy, followed by corticobasal degeneration. But we have also a small group of particularly rare primary tauopathies called globular glial tauopathy, Pick's disease, chronic traumatic encephalopathy, and a small group of patients who developed the disease because of a genetic defect in a specific gene called MAPT gene. And so what we are developing a digital image analysis collection where all the pathology slides, which we have prepared from these donations, and after they have been stained for these pathological dysfunctional proteins, we digitize them so that we no longer need to use microscopes, but we can actually see these pathologies on a digital screen. Then, once we see them on digital screen, again, using computational programs, web-based computational programs, we are selecting manually by drawing the regions of interest on these slides. And then what we are doing, again, using the artificial intelligence, computational tools, we have developed algorithms where we can quantify this pathology, either PSP pathology or this additional Alzheimer's or Parkinson's disease pathology, so that we can quantify this pathology in large scale, in an automated manner, so that we no longer need to rely on just on a human eye. And in addition to that, what we are also using the artificial intelligence for is learning, teaching computers to recognize the different patterns of inclusions in cells, which then are specific for specific diseases. And with this, we very much hope, and already are seeing, that enables us to diagnose the diseases and identify very specific patterns characteristic of unusual clinical symptoms in a much more accurate way than we were able to do ever before. What we are also doing, and here I'm showing an example from just over 200 patients with progressive subnuchal palsy, we are going through all the longitudinal clinical notes to try to identify whether there are specific patterns of a disease progression which would be characterized by specific clinical symptoms. And what is shown in this diagram that actually by grouping all this large collection together, we can identify specific progression patterns characterized by specific clinical symptoms, which are then characteristic of very specific clinical phenotypes of patients with progressive supranuclear palsy. Then by combining these longitudinal clinical data with this computer-assisted pathology quantification data, what we are then looking whether we can see any specific patterns which would again help us identifying where the disease started and in which patients the disease progresses more aggressively. And that we already are seeing that we can indeed identify. For example, in patients where we can see that in patients who has the shortest survival, those patients have the highest, this abnormal tau protein deposited specifically in a very specific brain region in the midbrain. And that will very much help in future to diagnose the disease early in using imaging as patients develop symptoms. The next thing what we are very much interested in doing and are collaborating with our colleagues from the computational science department, we are inputting these quantitative computer calculated pathology burden loads in our algorithms, which enable us then to see how, based on the different patterns of these pathological proteins, some of which are characteristic of primary disease, but as I already mentioned, oftentimes there are additional pathologies, how they differently spread across the brain and are characteristic of specific disease phenotypes, which we then can compare with pathology and clinical phenotype data with the genetic data, which we're also in parallel acquiring, and also then enables us to using other computational tools to model even more specifically to really understand in each individual what were the reasons why the disease progressed very quickly to very specific anatomical regions, so that we can understand how to treat these patients to provide sort of person-specific, individual-specific treatment based on the pathology propagation pattern characteristic for a specific individual. And what we already have identified using this computer-assisted pathology quantification and reviewing the clinical notes and identifying these unusual clinical phenotypes, that there are differences specifically in patients with cortical predominant phenotypes, such as corticobasal syndrome or language dominated phenotype or behavior variant dominated phenotypes, that there are indeed differences in tau pathology deposition in very specific anatomical regions. And as an example here, I will show you what we have identified in patients who present clinically with something called corticobasal syndrome, but pathologically they are confirmed to have PSP pathology. And that we see also in other atypical phenotypes, such as language-dominated phenotype. But as an example, I'll show here the corticobasal syndrome. So what we see here, that in these patients, consistently in this part of the brain, which is called the subcortical white matter, there is much more tau pathology deposition when compared with that specific anatomical region. So this is the parietal lobe, compared with other clinical phenotypes. And here, all these blue lines here show how in this group of patients with CBS clinical phenotype phenotype of progressive supranuclear palsy, how really that specific anatomical region, superior parietal lobule, which is here in green shown, how very much that tau pathology is much heavier when compared with all the other clinical phenotypes. And the next thing which we are particularly interested is using very advanced technologies called spatial transcriptomics to understand whether we can identify specific cell changes in this anatomical region where we see this significant increase in tau pathology load, whether there are cell-specific changes which could explain why the pathology developed in that region and caused that clinical phenotype, so that eventually we can use specific drugs to target those specific cell regions and cells and the pathological alterations in those cells to stop the disease development. And we so far have managed to show that there are cell-specific changes using a very small number of brain samples, which we now hope that we will be able to expand and prove what we already see in a very small subset of cases in a much larger cohort. So here, using this very advanced technology called spatial transcriptomics, what we did, we looked at specific anatomical regions, so different cortical layers and the subcortical white matter. We stained the tissue for astrocytes, these supportive cells in which we see the pathology most. Then we were able, as you can see here, select out specifically these astrocytes and do the gene activity analysis in these astrocytes in a specific anatomical region. And what we see that in these astrocytes, there are indeed, in patients with corticobasal syndrome, compared with the other phenotypes, there are very specific gene activity changes, which are very much involved in tau clearance and pathological tau uptake in these cells, really highlighting that there are cell-specific alterations which define the disease course and clinical phenotype in these patients. We also see that there are changes in different cell types called microglia, which is another supporting cell, but these changes are very different from those what we see in astrocytes, really showing that there are cell-specific alteration which drives the clinical phenotype. So the next thing, what we are very much interested in, this is what work we are doing due to the very generous funding from PSP Association, is to see whether we can identify in these characterized cohorts based on this quantitative pathology and longitudinal clinical phenotypic data, whether we can identify a disease biomarker which would then enable us to diagnose the disease early. And here, I need to explain a little bit more about this pathological dysfunctional tau protein. So we know that these different diseases, such as progressive supranuclear palsy or corticobasal degeneration, they develop because there are distinct protein filament shapes, which then accumulate in different parts of the cell body in corticobasal degeneration, in these distal processes, in progressive supranuclear palsy, in these proximal processes. And we know it's because of different conformation of this pathological tau protein. And that is one way how many people are trying to use it and try to detect this pathological conformation, this difference in the shape in fluids, in bodily fluids, to diagnose disease early. But we also know that there is another event happening in these abnormal dysfunctional tau proteins. So what's happening in these dysfunctional proteins, in addition to them changing their shapes, we also know that there are differences in how different chemical tags are added to these proteins. And these chemical tags, they have very fancy biological names, but they are different, how they are added to different pathological conformations of tau. And we specifically are interested in looking whether detecting these chemical tags, whether that would enable us to diagnose progressive supranuclear palsy or corticobasal degeneration in bodily fluids early on in disease process, because it's increasingly recognized that these different conformations are associated with these different chemical tags. And for that, I will briefly talk you through to explain what this whole process includes. It's a very, very novel technology, never used in human tissues before. And that's one of the main reasons as well why we are so very much excited, because we know that for DNA and RNA, it's already working really well. And that's why we are really excited to pursue this work and to demonstrate that it can also be done at the protein level. So for that, we are using either brain donation materials from multiple anatomical regions or the bodily fluid, the cerebrospinal fluid. So the brain regions we are homogenizing, so smashing them all up that they look very similar to a fluid material, such as cerebrospinal fluid. Then we are using antibodies, which are attached to metal beads. We are adding these antibodies, these metal beads, to the either this homogenate or cerebrospinal fluid. Then if there is this pathological tau protein, then it attaches to the antibody. And because the antibody itself is attached to this metal bead, then using a metal detector, we are able to separate these antibodies out from either cerebrospinal fluid or this tissue, homogenized tissue. Then we are adding special proteins which digest the protein in smaller bits so that the analysis can be performed. And then we are using this very, very novel technology called nanopore sensing, where there is a membrane with tiny, tiny nanopore size, really tiny holes in the membrane through which and these holes, they are not just simple holes, they are chemically designed probes holes, which enables to get this protein, this digested peptide, through at an individual basis. And whilst this peptide is going through this tiny, tiny pore in the membrane, then this membrane is also attached to a computer. And whilst it's going through this pore, based on these differences in these chemical tags attached to the peptide, it's generating different electrical currents. And these electrical currents, they can be registered on computer, and based on the changes in the electrical currents, which are influenced by these chemical tags attached to the protein, we are then able to separate out these distinct chemical tags, which are defining distinct misfolded proteins. And as I already mentioned, that's a very, very novel technology, which is actively used for detecting DNA, genetic alterations, gene activity alterations, but has never been used before for detecting alterations at the protein level. But there is a huge, huge potential for this being a novel diagnostic tool for diagnosing disease early on. And on this note, I will just summarize what is our passion in primary tauopathies and our work we are doing currently. So as I already I talked to you about, using computer artificial intelligent assisted technologies, analyzing the pathology data and clinical data, we are determining how co-pathologies in primary tauopathies could worsen symptoms at different speeds and be causes of unusual phenotypes. Using gene activity mapping in specific cell types in specific anatomical regions, we are looking whether focal molecular alterations are also responsible for unusual atypical phenotypes in patients with PSP or CBD. And using the nanopore sensing supported by PSP association, we are looking whether we can identify disease-causing protein modifications by checking these chemical tags to enable diagnose primary tau-pathologies early on in the disease. And as already mentioned, I'm working closely with Patrick Cullinane, who was the receiver of the PSP Association funding, and he had to be in clinics today, so that's why I'm here on his behalf. And very happy to take any questions if there are any.
Thank you very much, Zane. Just a high-level question from me to try and pull together. So the research you've been doing has been on the brain bank tissue, hasn't it? But what I think you're saying is that you are hoping to identify ways of measuring some of these alterations to proteins in fluids in people who are still alive.
Exactly. Either fluids or also skin samples. Skin
So it
samples
can
as
be
well.
from the skin. It doesn't have to be
Yes,
from the brain.
exactly, because we already are increasingly recognizing that even in skin, similar alterations occur, and therefore a skin sample could be also a potential way how we can diagnose these diseases early without requiring fluid or any other tissues.
Gosh, yes, that's a big step forward, isn't it? Once we can do that. There is a question about how much coordination there is with similar research projects around the world. And I guess linked to that, is there any kind of single database that's starting to gather together research on similar areas?
Yeah, that's a very, very, very important question. So Queen Square Brain Bank is a research facility and a resource where we provide the tissues for research across the world. And whenever we receive a request for research, the first thing what we are looking for to see whether we are already aware of similar studies going on, so that we can help to link the researchers together so that the efforts are complementary rather than identical and just on smaller cohorts. So that's one thing. The other thing is that we are also very eager to develop the database, as you mentioned, because currently, very surprisingly, it actually does not exist. There isn't a sort of an international, worldwide single database where all this information would be collated. But that's definitely an important thing which we currently are working together with another for Parkinson's disease. We are trying to develop such an international single resource, and therefore for primary tau-pathologies that should be equally important as obviously for other diseases. But yeah, that's one of our particular key priorities, what we are very keen to develop.
Okay, still a work in progress, but yeah. So I have a question. Can you define what a phenotype is?
Yeah, so a phenotype essentially means that the symptoms, there is a set of clinical symptoms. And for progressive supranuclear there is a characteristic set of symptoms which patients have. However, we know that there are patients who have unusual additional symptoms or completely different symptoms, not even anything typical of progressive supranuclear palsy, like falls or eye movement abnormalities, that they have actually very different symptoms resembling completely different disease, let's say Parkinson's disease or even Alzheimer's disease. And that means that these two groups of patients, they have different phenotypes. But under microscope, the pathology is the same. And that's exactly what we want to understand. What is the cause? Why these patients present clinically so differently? Because that's the main problem. That's why clinicians like Patrick, they struggle to diagnose the disease early on, because the phenotypes, the symptoms from one patient to the other can vary so significantly. And that means that we need additional diagnostic tools to separate out and identify these diseases early. Yeah, so the phenotype in a nutshell means a combination of clinical symptoms, which in one disease like PSP can be very different combinations, but pathology looks the same.
Thank you. No, that's useful to help people understand why it is so difficult to diagnose these diseases. Right. Well, we've come to the end of my questions. Megan, anything else from you?
No more questions in the chat at all.
Thank you so much, Zane. That was really interesting. I will definitely be watching it again because you packed a lot in, but it's really exciting what we're starting to be able to do with the image recognition power that we're getting now. So many thanks for sharing with us.
And thank you again for very kind invite.
Our pleasure. Right, so